Hello, I'm Gary Green. I'm the Chief Workforce Development Officer and faculty member at the Institute for Regenerative Medicine, and I serve as Chief Operating Officer for the RegenMed Development Organization. I want to welcome you to today's webinar, which is entitled From Curriculum to Workforce, Bringing Regenerative Medicine to Your Students. We are pleased to have the sponsorship of not only the National Science Foundation Advanced Technological Education Program for today's program, but also the RegenMed Development Organization, AlphaMed Press, who's the parent organization of Stem Cells Portal, and Bank of America, whose financial support helps make today's webinar possible. I want to, at this time, introduce our moderator for today's program. Bill Woodruff is the Professor Emeritus at Alamance Community College in Biotechnology. And Bill will be introducing our presenters and panelists and conducting today's webinar. Thank you for joining us today, and I hope you enjoy today's program.
Hi, I'm Bill Woodruff. Let me extend my welcome also to this webinar today, in which we'll explore the practical application of some aspects of regen medicine curriculum to the training of our future workforce. The term regen medicine obviously encompasses such a vast array of activities and fields, such as material science, 3D organ printing, things from stem cells to gene and cell therapy, tissue engineering, et cetera, it just goes on and on. Obviously, we can't cover all of that today, although we do hope in the future to use other webinars to cover more of these topics. But today we are going to look at 3 examples of the practical application of regen medicine curriculum to the training of our workforce. The presenters, all national known figures, include Dr. Cheryl Burrell, Department Chair of Biotechnology at Forsyth Technical Community College in Winston-Salem, North Carolina. Dr. Burrell's program is uniquely noted for its association with the Wake Forest Institute for Regenerative Medicine, in which her students are able to obtain not only internships, but often permanent employment as well. Also presenting today will be Dr. Margaret Bryans, professor and lead biotechnology at Montgomery Community College in Pennsylvania. Over the last decade, Maggie has been the principal investigator of the NSF-ATE Regional Center for Biomanufacturing, known mostly under the title of NBC2. She has developed both lab-based and didactic open source curriculum for training biomanufacturing technicians. And much of this curriculum can be found at the NBC2 website, biomanufacturing.org. Also presenting is Professor Jim DeKloe, who's had 30 years of developing biology, biotechnology, and biomanufacturing curriculum, including a bachelor's degree at his community college in biomanufacturing at Solano Community College in Vacaville, California. Jim has garnered numerous honors and awards and publications over his illustrious career. Those of us that have had the pleasure of hearing Jim speak in the past know that we are in for an informative and dynamic presentation. With that, I turn you over to the presenters. Thank you and enjoy. By the way, as we go along, if you have questions, you may submit them through the chat section.
My name is Jim DeKloe, and I teach biotechnology at Solano College in Northern California. And what I'd like to talk about today is developing a workforce for regenerative medicine. I especially like to focus on the development of a workforce to produce CAR T therapies and other immunotherapies, other cell therapies, and also to produce a workforce for the production of gene therapies. And as you can see on the left, there have been many recent approvals of cell and gene therapies. And so many of them are coming on the market. And on the right, you can see that in the near future, the growth in the number of these therapies is exponential. There are many companies working on them. 580 cell therapy companies in the world is one example, and many of them have products of various parts of the pipeline. And we're expecting many approvals in the near future, and we'll need a workforce in order to have those. This is part of the emerging $30 trillion global bioeconomy. And that bioeconomy will include pharmaceutical applications, and will also include non-pharmaceutical applications like commodity chemicals, like the production of fibers, the production of fuels, the production of new foods, using living cells. But what we're going to concentrate on today is the pharmaceutical side. President Biden in September signed an executive order, and a few days later, he had a subsequent summit. And that summit was looking at some of the white papers that were published in the last few years that predict a $30 trillion global bioeconomy. This is a really, really big number. The current GDP of the United States is $21 trillion. And so a lot of the economic advisors said, I went to the consultants, I said, check those numbers. They sound really big. And the consultant said, those are the numbers. And the regenerative medicine space is going to be a major part of that. We're looking at cell and gene therapy, and these are very complicated products, much more complicated than the protein pharmaceuticals that we are used to. They're going to be viruses, which are multiple proteins, that is much more complicated than a simple protein, and then the cells that are many, many times more complicated than even a virus. And the way this is shaking out is lentivirus is being used to transduce cells for cell therapy, and then the platform of choice in gene therapy is adeno-associated virus. So we have to train graduates to produce all of these therapies, or else we're going to run short, and some therapies may not come to market if we don't have enough trained personnel in order to make this a reality. In cell therapy, there are two major approaches for the production of CAR T cells. The autologous approach takes cells out of the patient, and these are usually really, really sick patients. They're a month away from dying, and this is often their last choice, their last chance. And so their immune cells are taken out and selected for the appropriate types of cells. The cells are activated. They are transduced, typically with a lentivirus. The lentivirus takes genetic information into the cell and has the cell produce and display the chimeric antigen receptor on its surface. That antigen receptor is specific for antigens that are on the particular tumor that is the target. This is put into a single-use bioreactor, divide. The number of cells are dramatically expanded. It's frozen and then mailed back to the physician, and the physician will put them back in the patient, and the cells will go around and destroy the cancer cells with a dramatic effectiveness. Many patients are cancer-free by the end of the month. The alternative approach is allogeneic CAR T production. In the allogeneic approach, immune cells are taken out of a healthy patient. They're genetically manipulated. They are dramatically expanded and then frozen in batches, and now you can utilize those more like a traditional product where you take a batch and you thaw it and then use it on a patient right then and there. In adeno-associated virus production, two main platforms are emerging. One is producing the virus using genetically engineered HEK293 cells, a human cell line. And in this approach, you transfect with three different plasmids, that is, three different sources of genetic information. And then those code for everything that's needed by the cell to produce viruses. And then the viruses are produced. You break the cells open with detergent, You take the viruses, put them through an affinity chromatography step, multiple filtration steps, and then the viruses are ready. After concentration and formulation, they're ready to be put into a patient. The other approach is producing these viruses using insect cells, using an insect cell line. And that is emerging as a major platform as well. So both of these approaches we're going to see, and we need to produce a workforce that is familiar with both. For stem cells, many of these are on the market. I have a student that had a treatment for her knees with human stem cells. And when I met her, she was in a wheelchair, and aspects of her walking were small. And now, after stem cell treatment, after Macy treatment, she now is fully walking and fully ambulatory. Exosomes are an emerging product as well. These are small membrane-bound spaces that contain a useful material inside, and these can be harvested and then used as therapies. We're going to need a workforce that knows what these are as well. Here's an article that talks about staffing challenges must be resolved if cell and gene therapies are to go supersonic. That is, if we are going to experience the major exponential growth that I've been talking about, we're going to have to dramatically increase training programs, dramatically increase the output of people who know how to produce these therapies. And here are some pictures. Over on the side is my student who had the stem cell treatments, and she's been inspired to go into stem cells. We are going to need people who are familiar with every phase of the development scheme of a drug, of the life cycle, and especially we are concentrating at my college at producing technicians who are familiar with producing products that have been that are being marketed, that have been approved by the FDA. We had a grant through NIMBLE, the National Institute for Innovation in the Manufacture of Biologics, and we assembled experts from around the country at all of the major hubs for the production of cell and gene therapy products. And we asked them, what's different about these products from the traditional protein pharmaceuticals like monoclonal antibodies that we're more familiar with, that we've been producing really since the early 80s. And here are some of the differences that they told us. Cells or viruses are much more complex than pharmaceutical proteins. Again, they're made of many, many molecules, and therefore there's an added amount of complexity. Aseptic technique and cell culture skills, mammalian cells culture skills are critical in the production of both. The equipment tends to be smaller scale and different. That has an advantage in training, so we can train on the real equipment. All equipment is single use. It's inside of plastics. In many cell therapies, especially autologous CAR T cell therapy, there's a time sensitivity. You're on the clock, a higher sense of urgency. The patient is waiting for this therapy, and you can't mess up or else that patient may die. In cell therapy, source material may be limited and variable. For cell therapy, quality control samples must be small since the amount of material is very limited and precious. All the material that you use to test is material that isn't ending up in the patient. Cold chain is critical. Chain of identity, chain of custody is critical. And then the quality control tests differ from those used in protein biopharmaceuticals. So we're going to have to train students on new techniques, especially flow cytometry. In manufacturing, this all starts with quality by design, which is an approach that isn't familiar often for people that are in research or early discovery, or even for those that are carrying out clinical trials. And so we have to teach the students quality by design, And there are some good resources to do that. Let me recommend these two resources. They were recently published last year from the Alliance for Regenerative Medicine and NIMBLE that I mentioned before. And these are essentially textbooks for cell therapy and for gene therapy. I use these extensively in my classes, and they cover everything that a student needs to know, easily downloadable, and again, free to the students. And here are the table of contents for this. For cell therapy, regulatory considerations, generation of quality target product profiles, risk assessments, cell collections, lentivirus production, manufacture of cell-based therapies, and analytical technologies. This is an evolving area, but this is hot off the press, state of the art. Likewise, for gene therapy, regulatory considerations, standards in gene therapy, also quality by design, process development, upstream and downstream, how to test the drug product, and how do you keep the process, every unit operation, operating in a state of control. So these are resources that I recommend and that I use extensively in all of the training that I do. And I wanted to end by talking about how important the generation of talent is for economic development. And in our area, in Vacaville, we have successfully attracted in the last year and a half several cell and gene therapy manufacturing companies. And a major thing that they were looking for is a trained workforce. And our college was able to provide that. And so for all of the communities who are looking to develop the production of these therapies in their communities, the establishment of a robust training facility in their community is a prerequisite. And thank you very much for your attention. We all have to get busy because we're going to need a lot of people.
Good afternoon. I'm Margaret Bryans from Montgomery County Community College. We're located in Bluebell, Pennsylvania, which is just outside of Philadelphia. I'd like to acknowledge the National Science Foundation for funding a project called Building an Advanced Therapy Technician Workforce in Southeastern PA and funding the production of this curriculum that I'm going to talk about today. I'd like to acknowledge Hetal Doshi, who's the grant manager, who helps me create the curriculum for the courses. The Philadelphia region is a hub for cell and gene therapy companies. There's 40 of these companies in Philadelphia itself, with many more in Montgomery County, as well as some larger contract development and manufacturing organizations such as WuXi and Charles River and Center for Breakthrough Medicines. These companies are hiring scores of technicians to manufacture cell and gene therapies. So advanced therapy bioprocessing technicians are in demand in the region and also nationally. Our associate level graduates are hired into roles such as aseptic manufacturing technician, viral vector manufacturing associate, for example. And from what we learned from developing the WeBet skill standards and also from conversations with regional hiring managers, our students should understand how to work in a regulated environment, understanding CGMP and good documentation practices. They should have a knowledge of single-use bioprocessing equipment and an understanding of the process of making a cell therapy or a gene therapy. So to address that, we're developing a new course in advanced therapies, and which will become part of a new certificate program. But in the meantime, I wanted to take the curriculum that we are developing and incorporate it into existing courses. And the 200 level biotechnology course seemed to be the perfect course to do this. So a couple of years ago, I started incorporating more gene therapy material into lecture, but also into some of the projects we were doing. And one of those was to use the gene therapy Zolgensma as a case study and have the students participate in a student research symposium where each of them in pairs would study a particular journal article that pertained to the development or the research of finding the genes responsible, creating a mouse model, and finally the successful clinical studies that led to the approval of this drug for spinal muscular atrophy. Within this course, I taught extensive cell culture, but we also taught transfection and microscopy and Western blots and separation methods such as chromatography and plasmid DNA preparations and analytical methods like STS, PAGE, and qPCR. So I realized I could take these techniques and put them together and develop an AAV production module and have a gene therapy focus to the work that we were doing in this research course. So of the 4 FDA-approved gene therapies that are currently marketed, 2 of them are adeno-associated virus-based, meaning the vector that takes the transgene into the cell is an AAV vector. These are Zolgensma, which I just mentioned from Novartis for SMA, and Luxturna. So Luxturna is interesting because it was developed at the University of Pennsylvania and it's currently manufactured by Spark Therapeutics right here in Philadelphia. The other 2 are produced by Bluebird Bio, Skyzona, and Zynteglo, and these are lentiviral-based recombinant viral vectors. Both types of vectors can be produced in HEC293 cells. So I thought it was important for the students to be introduced to growing these cells and learning how a viral vector could be produced using these cells. So this is the process that we want to mimic in the lab, in the teaching lab. So a process flow diagram for pilot or commercial scale AAV production. And as you can see, we're starting with our frozen vial of HEC cells and culturing the cells and expanding the culture, scaling up to a production bioreactor, which would then be transfected with 3 plasmids that are required for viral assembly within the cell. The cells are then lysed, treated with nucleases, and centrifuged to produce a clarified lysate, which is filtered, and the recombinant virus purified by affinity and ion exchange chromatography steps. These cells can also be grown adherently, but we chose to grow suspension cells. So in our module, we will have a very small cell culture volume at 30 milliliters, and we'll grow them in shake flasks and continue the process from this stage on. So this is the AAV production module that we're carrying out in the research class. So the students grow the HEC293 cells over several passages, and in the meantime, they prepare their plasmid DNA they're going to use for the triple transfection. This will allow them to introduce the required AAV and adenoviral genes, as well as our transgene, which is GFP. They'll grow the cells, lyse the cells, then analyze the cell lysate for viable recombinant AAV GFP. And to do this, we will transduce our target cell line, in our case, CHO cells, with the virus, and then analyze expression of the transgene. So these are the reagents that we purchased in order to do the experiments. We chose a suspension HEC293 cells, and we purchased it from Thermo Fisher. This actually very conveniently comes as a production system kit with the transfection reagent, the lysis buffer, the media, and the cells. We also purchased the 3 vectors that are required for viral assembly from Cell Biolabs, and this diagram on the right shows the 3 vectors that we use. Of course, we have the AAV vector with the inverted terminal repeats and our gene of interest, which is the GFP gene, the PAAV vector containing the rep and cap genes that are required for replication and capsid formation, and the P helper plasmid, which has the adenoviral genes that are required for viral assembly within the cell. And here's our 293 cells. So the virus is assembled within the cells are grown, and the cells lyse to release the viral particles, which are then purified and can be used to transduce a target cell and look for transgene expression. So in keeping with working in a regulated environment, we developed standard operating procedures for each step of the process, beginning with resuscitation of the frozen cells and culture of the cells in suspension, and cryopreservation of these cells to make our initial cell bank. So we did this work in collaboration with Izo Bayala from Quincy College. In the meantime, the students studied the components of the expression vectors and transformed bacteria in order to grow bacteria up to make sufficient quantities of the vectors to be transfected into the cells. So we see here the 3 vectors I described earlier, and the QC experiment for the students' plasmid preps, maxi preps that they did and compared to the commercial plasmids. So now that we're ready to move on to cell growth and triple transfection. So the cells are grown at 37 degrees C with 8% CO2 and shaken at 125 RPM. The plasmids are introduced using a lipid particle transfection reagent, incubated for a further 72 hours, and then the cells are lysed and centrifuged. So here's the cell suspension post-transfection and then post-lysis with the cell debris pellet and the clarified lysate containing the AAV GFP. This is a smaller version that shows a little more clearly the clarified cell lysate. And here's the SOP the students use to perform this part of the process. So at this point, the students wanted to find out if they had indeed made a recombinant AAV particle in the HEC293 cells. So we wanted to test the lysate for the presence of viable AAV. And to do this, we chose to transduce Chinese hamster ovary cells, so CHO cells. In this experiment, we'll look for expression of the GFP transgene by fluorescent microscopy. And these are CHO cells that were incubated with 2 different volumes of the HEC AAV GFP lysate, and you can see increased expression of the transgene and a little bit higher magnification. For control, we used commercially available AAV2 GFP viral particles, and here we have negative control with no cell lysate and the SOP that was used. We also wanted to look at transgene expression using Western blot, and so we repeated the experiment in the last slide, but this time incubated the CHO cells for 48 hours and then made a lysate of those cells and analyzed those lysates by Western blot. So what you see here in the first 4 lanes are CHO cell lysates that had been exposed, or the cells had been exposed to decreasing volumes of the HEC AAV, HEC produced AAV. And the blot was probed with an anti-GFP antibody. And here you can see levels of GFP expression decreasing relative to the decreasing amounts of AAV they were transduced with. And here is our positive control, which is the commercially available AAV GFP, and our negative control with no AAV virus. This is a antibody non-specific binding band that's binding to something in the CHO cells and not related to the GFP or the virus. So this analysis could also have been done using flow cytometry. This is an experiment that was done by Dr. Dominique Ngato, who is part of the WeBet team, and she's at Mira Costa College. So using commercially available AAV GFP, she transduced Chinese hamster ovary cells and studied the efficiency of the transduction by flow cytometry. So here you can see the negative control with very little population of cells, fluorescent cells. And here you can see the population of fluorescent cells here and the non-fluorescent cells in these pics here, indicating that the efficiency of transduction was about 85%. So we're currently developing the SOPs for our protein purification step, and then analytics for concentration and also purity by qPCR, HPLC, and ELISA, as well as STS-Page silver stain. So that's our AAV module that we've so far introduced into our research course. And I want to switch gears a little bit here and just tell you how we're addressing the aseptic processing and use of single-use technology. So at this time, we don't have our whole system set up as larger scale single-use. However, we wanted to give the students some experience working with single-use aseptic processing and aseptic transfer of media, for example, into a single-use bioreactor, in this case using a tube welder. And here we have one of our students who are gowned, and she's performing a media fill from a sterile media bottle into a wave type bag that the cells will then be introduced to using a tube welder, giving them that aseptic transfer experience. So all of the SOPs that we developed for this project are available to be downloaded on the MBC2 website at biomanufacturing.org. And here's the link to the curriculum unit for cell and gene therapy. I'd like to thank you for your attention, and happy to answer any questions.
Good afternoon, everybody. It is a pleasure for me to participate in this webinar entitled How to Bring Regenerative Medicine to Our Students. Like some of you on this webinar, I am also an educator. My name is Dr. Cheryl Burrell, and I am the Program Coordinator and Professor of Biotechnology at Forsyth Technical Community College. Forsyth Tech has been working to train students to enter the field of regenerative medicine for many years. And we have close associations and collaborations with Wake Forest Institute of Regenerative Medicine and other biotech companies focused on regenerative medicine in Winston-Salem and its environs. So as an educator, I do my best to introduce the whole reason for regenerative medicine and what students are learning in our labs and in our classes, how this can be applied to the field of regenerative medicine. So I explain to them that the goal of regenerative medicine, one of the goals I should say, is for scientists to grow cells and create tissues for use in patients. Now, in order for them to do this, they have to make sure that the cells and the tissues that they grow are structurally and functionally normal before we can introduce them into patients. And one of the ways in which this can be done is using immunofluorescence staining techniques. And this is what I do with my immunology techniques class, where I introduce students to both immunocytochemistry and immunohistochemistry techniques for them to have an idea of what scientists in regenerative medicine are doing. So I explain to students that immunofluorescence is a technique by which proteins in a cell or tissue sample, they are detected by fluorescently labeled antibodies, and these antibodies are specific to these proteins, so they bind to these proteins so that we can see where they are in a tissue or a cell organization. So we use, therefore, immunofluorescence to detect the protein, and also we can use it to detect the expression levels of a protein of interest in our cells and tissues. And this is what scientists can do in order to determine, is this tissue or cell behaving the way it's supposed to be behaving. So as I mentioned, we do a lab involving immunohistochemistry where protein is detected in the cross-section of a spinal cord. And we do immunocytochemistry where we detect protein in isolated cells. The cells that we use are Chinese hamster ovary cells or CHO cells. And we use these cells because they are pretty easy to grow and you can get quite a good number of cells in a short time. And students who do this immunology techniques course would have already done a course in cell culture. So they are quite competent in cultivating these cells because this is one of the cells that they work with in cell culture. So they already know aseptic techniques. And this is a good way for the students to practice the aseptic techniques again in this course because it's an important skill needed for the regenerative medicine workforce. So we use CHO cells, and what we're doing is to detect the levels of actin in CHO cells. And actin is a protein that is pretty commonly found across eukaryotes. It is highly expressed. So the likelihood of us getting levels of actin and seeing where actin is present in our cells is very high. So we choose these so that it's easy for students to do and be able to detect this protein called actin. So this is the overall protocol that we use. Of course, the protocol is more detailed, but in the interest of time, I'll focus on the general points of the protocol. And if you need more details, I am happy to share it with you. So cells are grown, CHO cells are grown, and then they are harvested and concentrated to the desired cell concentration per mil. And then after that, they are transferred to microscopic slides where a series of steps are performed when they're on the slides. For example, cells are fixed to maintain the structure of the cell integrity. And then the immunocytochemistry part of it is actually done. At the end of that, the cells are cover slipped and then visualized under the microscope. And the microscope that we use is an immunofluorescence microscope. Now, at Forsyth Tech, we have many good instruments, and one of them is a cytospin. And the cytospin is basically a specialized centrifuge that can transfer the cells from being in a cell suspension in a cytofunnel, which is seen here. We transfer the cell suspension, the cells that are in suspension here onto the slide, the microscopic slide, and the cells are transferred to a small area on the slide so that it's concentrated, so the likelihood of you obtaining enough cells for detection after this series of steps in immunocytochemistry is very high. So this has proved to be very useful to us. I normally explain the principle of immunocytochemistry and align it to what the students already know. So because this is an immunology techniques course, they already know about antibodies and how they work naturally in the body to defend us against harmful invading organisms. So they know already that antibodies are normally made against proteins like antigens like proteins, and they're specific, so they can bind specifically to that particular protein. So that helps in terms of eliciting an immune response to rid the body of that invader. So scientists, as I told them, scientists have over the years exploited naturally occurring phenomenon in our bodies to something that can be used to benefit mankind, and this is no exception. So scientists have found a way to commercially make antibodies against specific proteins, which would be the antigens in this case. So that's what happens. We can actually commercially purchase a primary antibody that is made to recognize specifically antigen. So we have our cells on our slides. We have the cells contain the protein actin. So actin is going to be recognized by the primary antibody, and it's going to bind. And then it's going to be left for a period of time. And then after that, the excess unbound primary antibody is washed off. And then secondary antibody is added. And the secondary antibody is commercially made so that it can recognize and bind to the primary antibody. And the secondary antibody, the beauty of this is that it has a fluorophore that is chemically bounded to it. And in this experiment, we use FITC, fluorescein isothiocyanate, as our fluorophore, so that when it is exposed to UV light, it will fluoresce green. So the green in the image that you take on the immunofluorescence microscope will detect where actin is present. And this is just a picture that I took from one of the students, the experiments that student did. And if you notice the green, you can see that indicates the presence of actin. And we also stain the nuclei so that we can see where the actin levels are with respect to the nucleus, so we can have an idea of where the cell is. So the nucleus is stained with another fluorophore called propidium iodide, which is going to fluoresce red. So you see a good contrast between green and red here. And this is a very good, straightforward protocol to start students off with, to get them familiar with the whole principle of immunofluorescence by way of immunocytochemistry. And it's easy to do. So students like this, and it also helps them because if they go into the workforce, if they go on internship, for example, and they have to do an immunocytochemistry procedure, they will remember that they did this before. So it will be something that they're just going to build on their expertise before that they had learned before. Now, if you're at an institution and you don't have an immunofluorescence microscope, you can still introduce students to histochemistry or immunocytochemistry. In this case, the secondary antibody would not have a fluorophore attached to it, but it would probably have an enzyme that can break down a substrate that is visible if you use a regular microscope. And that is what we do for our immunohistochemistry lab. We're detecting the levels of dopamine hydrolase in the cross-section of spinal cord, and we get these cross-sectional tissues from Wake Forest Baptist Health, and we can use these tissues that are already on slides, and we just stain them, and the students can see them under the microscope. Now, this is not a picture that I got from students. This is just what I got that accompanies the antibody spec sheet. So, but you can have a picture looking like this if you were to do an experiment like this. So we have a number of people who we partner with in order to train our students to become competent in the workforce. And at this time, I just want to say thank you to them. I'd like to thank W Firm, Wake Forest Baptist Health, and other bioscience companies in Winston-Salem for the work they do with our students and for our students to help them to pave their way for them to be successful in this regenerative medicine field and also further on in their careers. I also want to thank the faculty and staff in the Life Sciences Department at Forsyth Tech for their contribution to training the students also. And of course, last but not least, I'd like to thank my biotechnology students. It's really a pleasure working with students and contributing to training them to go to the next phase in their life. A lot of them, they're patient with me because I try new things with them in the lab. They have a willingness to learn, and I push them to as far as they can go. And I'm just thankful to be a part of this. Thank you for listening.
All right, then I guess we're ready to proceed. I'd like to welcome you to the live question and answer session. First of all, I want to take my own personal opportunity to thank you all as presenters. Those were fantastic presentations. A lot of information shared there. I'm sure our audience is very much going to be able to utilize that information. So as we start,
Bill, can I say something before we start? So
absolutely.
I want to underscore this point. This field is going to be huge. And so we need to recruit many, many students into this field, and we need to expand the number of programs like Maggie's. She's a national treasure, and I was so inspired by what she's doing, and think of the generosity of her giving all of her SOPs out for free across the country. But everyone has to get busy. This is going to be huge. And at the same time, I've found that the students are really inspired by this field because we, for the first time, are talking the C word. We're talking a cure for cancer. We're talking about alleviating human suffering. We're talking about alleviating the suffering of children who are currently untreatable and have inherited a metabolic disorder through no fault of their own. I also would like to give a shout out, Maggie did, to the National Science Foundation and their support over the years for building this community of faculty that are developing and then exchanging these protocols and exchanging these experiments. It started with BioLink and now Innovate Bio, and also with the support of Maggie's NBC2. And so the National Science Foundation has been with us all through this. And then shout out for Nimble for their funding, and also for Biomade, building a workforce. This is an important work that we're all part of, and we're building this community. We're kind of building the plane as it's flying, and I wanted to make that point. And thanks, Bill.
Thank you, Jim. That's an excellent and, of course, a very valuable point. Maggie and Cheryl, do either of you want to add on anything now that you've had more time to think about stuff?
Oh, thank you, Bill. I just want to reiterate what Jim's saying. We're having employers contact us for graduates commonly now. I don't have enough graduates for the number of jobs in this sector. So it's a great place to be, but we have to expand our programs and keep up with the curriculum so that we're training our graduates as best we can for working in this sector.
Thank you.
I would say the same thing. Right now, we're having a lot of people contacting employers, contacting for students, and we can't get enough students. And we really need to do something about going into the high schools, going out and advertising, trying to find more ways of spreading the word through social media, because that is where a lot of people are going right now, just to get the people in to fulfill the needs of the workforce.
Thank you, guys. That's a very critical point, that when you're thinking about your own programs and you're trying to expand, that this is a field you can expand into. And there are many, many hubs around this country where regenerative medicine is really starting to take off. So we really look forward to the opportunities that we can open new programs, new curriculums, and get ourselves a good trained workforce. All right, let's take a look and see what we have for questions. We have one from Sarah. I believe this one would be to you, Maggie. How many weeks does the AAV module take?
Oh, okay. Thank you for the question. It takes about 4 to 5 weeks for a 15-week course. It's 4 to 5 of those weeks, and we meet twice a week for 3 hours. So it's paced quite well as far as, growing the cells and what we have to do with the cells to produce the virus. And then the analytics comes at the end of that. So within about 4 to 5 weeks, we can do the entire module. In the meantime, they're working on our case study and reading articles on, a gene therapy and getting the full big picture of gene therapy therapeutics.
Let me stay with you for a minute, Maggie. It's related. I have a couple of questions here relating to your HEC293 cells. One is from Sandy. How do you validate the HEC293 cells or any other cell line you might use? Oh,
we don't do a whole lot of cell line validation. I know that's very typical in industry. They know everything about the cells genetically and otherwise. We purchase those cells and they grow fantastically well with the appropriate media. So we've just gone off and running with that cell line. In other courses, we do, mycoplasma tests. We try to teach the students some of the work that we do to qualify a cell line. But for these particular cells, we just actually adopted them and started producing.
And Bill, let me add that, remember, HEC293 cells are biosafety level 2. So in order to use them, you have to go through a few protocols. It's nothing big, but you have to formally designate your lab biosafety level 2. An alternate to that would be to use the insect cell platform, which is biosafety level 1. AAV itself is biosafety level 1. So there are some additional hoops you have to go through if you use the HEC 293 platform.
And just to add to that, Jim, it wasn't a huge amount of changes to our lab safety protocols. We actually put a proposal together. We sent it to our college safety officer and our dean and said, these are the CDC guidelines. These are other guidelines. This is what we're doing. Sort of a assigned space with biological safety cabinets and restricted entries, some signage, extra PPE, and bleaching everything we use before it goes into the biological safety waste. So a few things we had to do, but it wasn't overwhelming to get that approval.
All right, to carry on, the follow-up with that one, though, is when you're saying to get the approval, did you have to go through your college to get that approval? And what was the process? Is there like an IRB or? For using BSL-2 cells? Yes.
No, actually, our microbiology program already have a couple of BSL-2 organisms that they use for training. So again, it's just taking what we had in our lab and adding those components I just mentioned and putting that proposal forward to our dean and safety officer and getting that approval. And that was sufficient. We actually wanted to do BSL-1 with the insect cells, but those vectors, the AAV GFP vectors, are not commercially available. So we went with the HEC. At some point, when they become available, we would like to grow the BSL-1 SF9 cells, but this time they were not available.
Very good. How about you, Jim? Did you run into an IRB issue?
We are using insect cells.
Oh, okay, very good. All right, so Cheryl, we have a question here for you, and one of them was, what kind of challenges did you face when you were trying to incorporate regen medicine into your existing curriculum?
So one of the challenge is that, as you all would know, that sometimes the processes can be pretty complicated for students who are beginning their journey into the bioscience industry. Sometimes they may not be aware of terminologies that we use in science. So it's a way to bring something that would be very complicated to a level that they can understand. And what I have done is to repeatedly introduce different terminologies as we discover them in our courses and relate them back to what the field, the application in the regenerative medicine field. And that is why we start with those basic experiments so that they can build off of what they know when they go out on internship. And another challenge that probably other institutions may face is not having enough resources to do some of these experiments, or they may not have the expertise. However, there are grants that are stipulated, especially in the area of Winston-Salem, where faculty are being trained both in the high school and in the college, at the college level, so that we can learn various techniques and then bring them back to the students so that we can properly prepare them for the workforce. For example, in the summer, I'm going to be at WFirm working on an externship there to learn more about the processes that they have there so that I can make sure that the students are properly trained for the workforce.
Thank you very much. Let's see, what do we got? We got a question from Sarah. I assume this would be for you, Jim. When will the biomanufacturing campus open? It sounds amazing.
The campus open. So we have a biotech training facility here in Vacaville that opened in 2017. We have 4 biomanufacturing suites. We have a cell culture suite with 14 biosafety cabinets. We have isolated rooms. Wow. And we have a bioreactor suite where we have, I think, 14 bioreactors. And then we have a recovery suite with multiple filtration and centrifugation systems, and then we have a quality control, really a run-of-the-mill laboratory on the end. So if that's what we're talking about, then we are open and have been operating since 2017.
Excellent. That sounds really impressive. How many students do you have in your program?
So as you said, Bill, we have multiple certificates. We have an associate's degree and we have a bachelor's degree. So we have about 50 in each of those components.
Wow, nice.
And what has been really gratifying is we have had really good relationships. We've formed really good relationships with multiple graduate schools and also with our high schools. And so if you put all of that together, we have articulated high school programs, stack certificates, add general education, and it's an associate degree. It flows seamlessly into our community college bachelor's degree. It's in biomanufacturing, not biotech, but biomanufacturing. And then that goes into graduate programs. So it's a education pathway from high school through PhD with multiple entry and exit points.
Thank you. Cheryl and Maggie, how many students do you all have?
Currently, I have about 30 students in the program right now that are active, and we are finding that we need more students because we need more people for the workforce.
Very good.
We have about 80 students in the program at various stages at the moment, but we could use more.
Yeah, these are sounding
right,
like
Bill, I think I could quadruple and place everyone. It's just there is not an awareness, and this is on the national scale, there's not an awareness of the opportunities in this field. There isn't an awareness that this is a place where students can get a career, not a job, but a career, and the career pays a livable wage to start, but provides a tremendous opportunity for upward mobility. I was at the UC Davis GMP facility on Monday, and the new director of that facility started out at the lowest entry level and moved up, and now he's director of the facility. So there's tremendous opportunity for upward mobility, and we have to get that message out nationally.
Okay, thank you. Looks like we might have time for just one more question. This is from Emeritus. It says, do you have opportunities to collaborate with your CGT companies as advisory board members, as experts on syllabus upgrade, training for workshops, or as interns? So I'll throw that out there for any of you that wish to respond to that.
I will. Yes, absolutely. I have an advisory board for the program, but I also have a cell and gene therapy group of educators and hiring managers that meet twice a year. This is part of my NSF ATE grant. And the information we get from them, the opportunities for students to have them at the table, engaging with us and knowing what we do is invaluable. So yes, lots of interaction.
Excellent.
And let me add to the Nimble grant that Maggie and I worked on with others across the country. We had a very, very interesting process where we met in the main cell and gene therapy hubs in the United States. We collected experts in Seattle, in the San Francisco Bay Area, in San Diego, in North Carolina, in the Philadelphia area, and in Boston. And we asked them, how does this field differ from traditional production of protein pharmaceuticals? And those experts gave us tremendous information, and all of that we wrote up in a report, and it is available at Maggie's website at biomanufacturing.org.
Excellent.
And at Forsyth Tech, we also have our advisory board meetings that we have yearly. And right now we're entering a phase where the employees actually drive our meetings so that we know firsthand what is needed in the industry. We do collaborate with Wake Forest and other institutions around here and the National Center for the Biotechnology Workforce. So we do have a great support system. We just need to know how to get the word out to get people in our program so we can produce the workers that are needed.
Thank you. And that seems to be the end of our time today. I want to thank our panelists. Also, I want to throw a shout out to our support from the NSF as well for all the things that we do, including the seminars or webinars like these.